So once again, good morning to one and all. And I think I have already introduced myself. So I think this is the eighth day of your training program now. And during the last week, you might have learned a lot of techniques, like NGS as well as the mathematics analysis from the AU team. And they have covered the uh, Illumina platform, Illumina MySig. Okay. So here in GBRC, you will be going to learn about the ion torrent technology, which is a semiconductor-based sequencing technology. So goal for the day is why we are doing DNA sequencing, who invented all these techniques, all the technologies that are available, especially for the next generation sequencing technology. I think Koringa sir and AU team already have introduced about the first generation, second generation, as well as the third generation sequencing technology. So I will not go in detail in this and the advantages and the limitation of the ion torrent technology. We do have both MySec as well as the ion torrent platform, but you have learned the MySec platform over there, that is Illumina platform. So here you are going to learn about the ion torrent technology. So this DNS, it was a very just brief history about like it was invented by the Patrick Meisters, that something is there in acidic properties in the nucleus of from the cells. And then these three scientists, Oswald, Avery, McLeod, and McCarthy, they finally proved that yes, previously it was thought that the protein is working as a hereditary material. But these three scientists have proved that it is not the protein, but the DNA is the genetic material. And then everybody knows that in 1953, Watson and Crick, they have uh, developed a model for the DNA double helical structure. So if you consider the sequencing, then DNA sequencing was not the first sequencing. People have sequenced the RNA first time, and it was the fast MS2 bacteriophage. It was sequenced first time in the world, and then DNA sequencing came into the market. They have sequenced DNA by converting RNA into cDNA, then they have sequenced, but it was the RNA virus MS2 they sequenced first. In the first method for determining the DNA sequencing, it was developed by the Ray et al. And the Professor R. Padmanabhan, who is an Indian scientist from the Cornell University, and they have sequenced the, this fast as a cohesive ends, some 25, 27 base pair. So they have first time sequenced the ends of this bacteriophage, cohesive ends, using the primer extension, specific primer extension technology. And this technology then was adopted by the Sanger, this Frederick Sanger, by using the dideoxytermination method. So it was the this Ray who developed the DNA sequencing technology, and it was later adopted by the Sanger sequencing technology. Sanger. Later on, not later on, but simultaneously, this uh, uh, Sanger and Megzam and Gilbert also developed the same technology. However, the principle was different. So initially, if you look at the era of the sequencing, most of the things was semi-automated. Many of the things you have to do manually, and later on, we can see that this ABI 370. It was the first fully automated DNA sequencer available in the market, developed by the Applied Biosystem. And then in 2000, then this is the era of the first generation. This Sanger sequencing is a gold standard method. It will remain there. Okay, so starting from here, from the development of the ABI 370 first DNA sequencer to till now, first generation sequencing is still going on. Then this is the era of the second generation sequencer. Then the third generation sequencing, like the Tech Bio and the that nanopore, Oxford nanopore and the smart technology. So today not only today, but during these 2 days, we will be focusing on this part only. And see, there are lots of sequencing technologies and platforms are available during this 2 days we will be covered only this ion torrent technology. So if you consider the first generation, Sanger and Exam Gilbert technologies was the first generation sequencing. Then the second generation, it covered the pyro sequencing, sequencing by synthesis, Illumina is doing sequencing by ligation, and the semiconductor based sequencing. Ion torrent using the semiconductor based sequencing. And if you consider the third generation, it will be smart, nanopore, DNA nanopore, and the other live strip sequencing technology. But these are yet not some of the technologies yet not mature because of the accuracy. You might be knowing that AUT have told them. So this is the just overview of the dideoxy chain termination method, Sanger sequencing, gold standard method. I'm not going in detail, but what important is the accuracy and the throughput. Because we know sequencing technology has accuracy as the Sanger sequencing technology, and that's why it is the gold standard method. It is considered the gold standard method. But the only limitation is throughput. If we consider again the advantage of Sanger is high accuracy and the read length. Read length is 800, 900 base pair. No second generation sequencer will give you this much of the read length. This is so the accuracy is very high as well as the read length is very high, but only the drawback is the throughput. In a single run, you may have only the 96 sequences only. If you run gold flat 96 sample. If you consider the next generation sequencing technology, sorry. So if you consider that human genome project, it took about the 13 years to complete with this many $1 billion to complete the single human genome project. So we know that the sequencing cost is getting reducing because of the high throughput technologies developed right now, and simultaneously the cost for human genome is also getting reduced as per to 2021. And because of this, the number of the sequences in the database getting increases because the sequencing is becoming cheaper. People are doing a lot of sequencing. So if we compare the Sanger and the NGS, so I have covered this. So I will go to NGS. So like here completed the human genome project. But now we know that we can in the with the NGS we have to just prepare the library and sequencing of that library. We can do multiple genome. Here single genome take about years. Here multiple genome can be completed within days or hours. And here you will get a few sequencing in a single run. Here you will get the millions of the sequencing in a single run based on the platform or the chip or the process you have used. These are the basic workflow for the NGS. So, by the DNA or RNA extraction, followed by the library preparation, and then the sequencing. And the differences come at here in 454, but the machine is not absolute. And in the iron torrent technology, they are using the immersion PCR. What is I will come into later. So they are using immersion PCR to amplify, clonally amplify the DNA fragment on the beads and then go for the sequencing. Like this part you have learned in the AAU, in the Illumina, they are using the bridge amplification for the cluster generation, and then they are using the sequencing by synthesis technology. So here they are in the Illumina, it is the cluster generation, bridge amplification, and in iron torrent, it is the immersion PCR. So QC of the library is mandatory. Okay. So because each platform has some limitations, like the read length, there should be shorter reads. Otherwise, it will create a problem in the cluster generation, or you may get the polyclonal. What is polyclonal? That will come later in session. So you can use bioanalyzer for the quantification. You may use the qubit or the real-time PCR or the digital PCR. Anything you can use. So now coming to the ion torrent sequencing technology. So it is using it is also called the semiconductor sequencing technology. So we know that during the DNA replication, one nucleotide is get incorporated, so one hydrogen ion is released. Okay. So here they have developed a technology which, when the hydrogen ion is released, there will be a change in the solution. Okay, so this technology is based on the detection of the hydrogen ions that are released during the DNA polymerization, and it is again the sequencing by synthesis chemistry only. And this was licensed from the DNA Electronics, and this platform, PGM, Personal Genome Machine, it was available in the from the Iron Torrent technology. It was IP to, during the 2010, and it was available in the market during 2011. So how this Iron Torrent technology works? So polymerase incorporate nucleotide into the growing chain, and each time it will when it will incorporate nucleotide, it will release a hydrogen ion, and these hydrogen ion will be detected by the uh, this ion sensitive field transistor. So there are ion current sequencing technologies uses a chip. So I will be drawing this. So for example, in PGM, Personal Genome Machine. So it was the first machine by the Iron Torrent technology they have released, but now they have stopped the support for this machine, and it supports the three types of the chip: Iron 314, 316, and 318. Then Iron S5, they are using the Iron 510, 520, 530, 540, and 550 chip. Each chip has its own that output. Using the different chip, you will be having the different types of the output. Like in Illumina, you will be knowing that Illumina MySec, reagent V2, V3. The only difference is the throughput. So here also the difference is the throughput. Here you will be getting some MB of the data. Here in the here 1 to 2 GB, in the 380 like the 2 GB of the data maximum is possible. Ion proton chip, it is a maximum capacity about to 60 to 80 million reads in a per run, while in 5 if you consider the 530 chip, then it is only 12 to 15 million reads. And another is Genexus. It is the latest version of this ion torrent technology they have developed. It has a 4 lane like Illumina. If you consider the Nova 6, they have a flow cell like the 2 lane and 4 lane. In each lane, you can run a different type of the library or different type of the sample. Here also, with this 4 lane, you can use single lane at a time or 4 lane at a time with a different type of the library. Like in a single lane, if you want to run amplifier sequencing, here you want to run short-range sequencing, here you want to amplifier sequencing, everything is possible. And the throughput of each lane is 12 to 15 million reads. So if you run a full chip, you will get about 60 to 80 million reads in a single run with a different type of the libraries with different applications. So this is the chip design that how it detect the hydrogen ion that are released during the incorporation of the nucleotide. So this is the so consider this as a chip, and it contains a millions of nanowells. And each nanowell there will be a single piece that I come to later what it contains. And everything DNA that sequencing is going on this well only, and beneath this well there are billions of nanosensors, ion sensitive field transistors are they have like that. So when uh, when there is a flow of the nucleotide, and when there is a complementary strand there, it will ligate the nucleotide, and simultaneously hydrogen ion is released. In those wells, there will be a change in the pH. It will be detected by these sensors, nanosensors, and it will tell that yes, your pH is there, and this nucleotide was incorporated. So first step is always library preparation and QC, and the second step is immersion PCR. So why we need to do the immersion PCR? Why we need to do the clonal amplification or the base amplification? Anyone can tell?
So that more change in ions will be there in that cluster
Yes.
because this technology deals with cluster generation. So
No, this Illumina is using cluster generation. Here it is immersion PCR. But Illumina Illumina does also require the cluster generation of the same fragment. The same fragment will form a cluster because in Illumina also it will detect the light by the camera. So if there will be a single strand of the DNA and one nucleotide will be incorporated, which is ligated by the fluorophore. So the light which will be emitted by the incorporation of the single nucleotide, it will be very minute. So camera will not detect it. So what they are doing the clusters of the same fragment. It will be multiplied by many times. So in a single cluster, there will be a single DNA molecule which get amplified hundred thousands of times. So in each cluster, there will be a incorruption of nucleotide. Same nucleotide will be incorporated in a particular position, and a light will be emitted. So it can be detected by the camera sensor. So similarly, in this technology, so suppose DNA K strand है, there is a single strand of the DNA, and if you incorporate a single nucleotide, one hydrogen atom will be released. So think how much pH change it will do. One hydrogen will nothing. It will be not detected by the even this nano sensor. So they are using this immersion PCR. Here the beads is there. It is called the ISP, ion sphere particles. So like in Illumina, they have flow cell, and flow cell is preloaded with the oligos. And that oligos are complementary to the adapter sequence in our library. Okay. So here also these ISPs are preloaded with the oligos. And that oligos are the complementary with the our library adapter sequences. And emulsion PCR means it is a mixture of water and oil. It will form a micro reactor. So the idea is that in a single micro reactor, this single micro reactor, it will contain a single ISP, one fragment of your library, single fragment of your library, and all the chemicals like the DNA polymerase, nucleotide, and buffer, which will require to carry out the PCR reaction. So, so now this is the ideal. If we consider this is ideal condition in a reaction, this micro well, sorry, my nano drop, we contain the ISP and this your target library DNA molecule. So during PCR, it will amplify this molecule many times during the immersion PCR. Like यहाँ पे PCR होगा, एक fragment यहाँ पे bind हुआ, it will PCR again denaturation. Then the second strand will bind with the complementary oligos or the ISPs, and likewise. So it will create a thousands of the copies of the same DNA molecule. So now this ISP ion sphere particle containing the target molecule which is amplified, it will go into the this bead, sorry, this valve. And this chip is designed in such a way that in single valve only one microbit will go. So the now here the sequencing will occur, and here there are several copies of the same DNA molecule. So lots of hydrogen ion will be released, and it will be detected by this ion series, this sensor.
But what is there in that ion sphere particle?
Sorry,
What is there in that ion sphere particle?
ion sphere particle it contains the oligos which are complementary to the adapter sequence in our library. Because in library preparation, first step is for example, we are doing the solvent sequencing. So first we randomly segment the DNA, either enzymatic sharing or the physical sharing. Then we attach then we library adapter. Because in sequencing, see, there are millions of the DNA fragments. We don't know the sequence of the end of that fragment. So somewhere we have to start now. So for that we are library adapters. Either it is same in both Illumina or the ion Torrent technology. We have to library some known sequences. Yes. And because these adapter sequences are known, during the sequencing, we are adding the sequencing primer in both the chemistry, either Illumina or this. These sequencing primer are complementary to this adapter, and from that it start the sequencing.
So in just in the luminary of flow cell in that oligos are
In
there. So
flow
instead
cell
of
also
that,
oligos are loaded.
But in iron torrent,
Ion sphere particles
so
are
that
there.
it will PCR will
Yeah, PCR will happen, and it will amplify the same DNA molecule, and it will form a ISP, ion sphere particle. In Illumina, it will form a cluster. So in now the this is the ion 1+2 system is there for the to carry out the emergent PCR. We will be demonstrating all these things in the hands-on. Now the second step is to enrich the ISP. So like यहाँ पे बहुत it is a probability like during the setting of the emergent PCR, we are reading the ISP and we are quantifying our libraries and we are mixing both there. So idea is that the basic idea is that there will be one ISP and one DNA fragment molecule. Okay, likewise we are mixing both the things in the during the emergent PCR. बट हर बार ऐसा तो नहीं है कि डाला है तो वो सब हो ही जाएगा। कुछ जो बीट्स है इसमें कुछ रिएक्शन है, कुछ हुआ ही नहीं होगा क्योंकि वहाँ पे कुछ डीएनए का फ्रेगमेंट नहीं गया होगा। ठीक है, तो वो अगर अगर ऐसे जो बीट्स है जिसमें कोई डीएनए का फ्रेगमेंट्स नहीं है, वो बीट्स अगर इस वेल्स में चले जाएंगे तो इट विल ऑक्यूपाइड अननेसेसरी वेल्स एंड वहाँ पे कुछ सीक्वेंसिंग होगा नहीं एंड यू विल गेट द less number of the sequences at the end of the run. So we have to remove those ISPs which do not contain any positive reactions. So machine, this ion enrichment ES system is there. We have to just provide the reagents and our emergent PCR product. It has a robotic arm and program is already set in the instrument, and we have to just provide the reagents and our samples. It will do the enrichment of the ISP, which is having the that our polyclonal amplified molecules. Third step is chip loading, which is very crucial. Like in Illumina, you have not to worry about the loading because you have to just provide the cartridge and your reagent. Machine will do the loading and the that cluster generation. Here we have to do these things manually, chip loading, because and this is a very small area. In chip, you will see that it is a very small area and it contains the millions of that nanowells. So if there is a minute air bubble will be there, then it will reaction will not happen there because of the air. And this is the process that how we have to just manually load our sample into the chip. We have to centrifuge and then we have to look at if there are any air bubbles or not there. And this after chip loading, this chip will go into the machine directly. So now they have developed the ion shelf. So advantage of the ion shelf is that everything is automated. Some of the library preparation workflow are also there, like basic library preparation. You have to just provide your sample like the DNA or RNA, and it will prepare the library. It will do the all the quantification. It will do the emergent PCR and chip loading. Directly chip load करके यहाँ को दे देगा ion shelf. We have to just provide the respective reagents in their respective fields. It will do all the things automatically.
Machine will do it automatically.
Machine will do automatically. We have to just tweak the program. But currently, this machine is supporting only ion that ion S5 system only. So we have both ion S5 and ion S5 plus. Both the system are there. We also have the ion shape system. I think I have already explained this thing. So now coming to the different chip and its throughput like ion PGM, the 314, 316, 315. So see the number this output ion 3400 MB data only in a single chip here up to 1 GB data and in 318 up to 2 GB of the data. Like in ion proton, this is the highest output of the ion torrent platform, like 60 to 80 million read in a single run. But only limitation of this ion proton is the read length, 200 base pair chemistry. the rest of the chip like in PGM or the INS5, they are supporting 400 base pair chemistry. But iron proton is supporting only this 200 base pair chemistry. So INS5 Gene Studio is five different types of the chips are available. 510, 20, 30, 40, and 15 with a 550 is the highest output, 100 to 130 million reads in a single run. We are using 520, 30, and 40. We are not using 550 and 10. So only difference is the chip size is all same in all. The only difference is the number of the nanovels in each chip, and that's why the throughput is different. And read length support is up to 400 to 600 respect. This is the GeneXA system. I mentioned that it is using the everything is integrated in the machine. We have to just provide the our samples and it will prepare the library. It will do the. It is not the iron chef. It is the GeneXA system, another latest machine that are in the market by the iron Torrent technology. Support the starting from the E- even the DNA extraction is possible here. You have to just feed the sample, it will extract the DNA, prepare the library, sequencing, and data analysis. Everything is integrated. We have to just 5 to 10 minutes hands-on time. You have to for the strategy program. What library you want to do with that reagents. And it has already told that it supports the 4 lines. And each line has capacity of the 12 to 15 million. So advantage of this is not second generation, but advantage of ion torrent or the semiconductor sequencing technology, high throughput technology, rapid sequencing speed, and the operating cost is somewhat less, and the avoidance of modified nucleotide because in Illumina or in smart technology, they are using the modified nucleotide having the fluorophore tags. Okay, so here it is not required. And the optical measurement is also not required because it is not using the that level nucleotide. It is detecting the pH. However, the limitation are difficult to read the long repeat, like the homopolymer stretch. Like in Illumina chemistry, you might be knowing why it is accurately read the homopolymers. How it accurately read the homopolymers.
Because one piece will incorporate, then it will be blocked.
Yes.
Others cannot come.
Yes, in Illumina, what happens when one nucleotide is there, it will be blocked, and then second nucleotide will be getting promoted there. But here it is not possible because when the nucleotide will flow, there will be a 5 or 6 of T in the your DNA strand. So it will add A in all that, and the pH change will occur. Likewise, pH change will also occur. So the sensor will not accurately detect. It is because the pH change is because of the five incursions of 5 or 6 nucleotides at a time. Up to 3, 4 strength, it is okay. Machine will detect accurately, but beyond 6 or 7, it is confusing. Second, shorter read length as compared to third generation sequencer, because third generation sequencer are giving reads in the number of kbs, and throughput is currently lower than the other high throughput sequencer. Like if you compare NovaSeq, Illumina NovaSeq, so it has a capacity to 2-3 TB of the data in a single run. So this is the just comparison table like sequencing platform with sequencing technology. They are using maximum read length possible, maximum output, accuracy, and cost. Thank you. If you have any questions. So now we can move for the hands-on training because we have less time. We have to do many things today. We have to quantify the library, then we have to do the what? Sorry, that we have to do that. We have to set emergent PCR by 5 or 6 o'clock. So if you have a, I think I can run this short video for 3 minutes.
Next generation sequencing is a powerful technology which can interrogate many targets at the same time, from just a few genes to all of the individual nucleotides in a whole genome. Ion Torrent technology from Thermo Fisher Scientific takes an entirely new approach to next generation sequencing, making it massively scalable, faster, simpler, and more affordable than ever before. The sequencing of DNA is done using a semiconductor chip, similar to the chip found in your digital camera. While the chip in your digital camera has a sensing layer covered with millions of pixels that translate light into digital information, an ion chip has millions of wells covering those pixels. These wells capture chemical information from DNA sequencing and translate it into digital information, or base calls. The sequencing process starts when a sample of DNA is cut up into millions of fragments. Each fragment then attaches to its own bead and is copied until it covers the bead. This automated process covers millions of beads with millions of different fragments. These beads then flow across the chip, each depositing into a well. Then the chip is flooded with one of the 4 DNA nucleotides. Whenever a nucleotide is incorporated into a single strand of DNA, a hydrogen ion is released. This is how the ion Torrent system sequences DNA. The release of the hydrogen ion changes the pH of the solution in the well. An ion-sensitive layer beneath the well measures that change in pH and converts it to voltage. This voltage change is recorded, indicating that the nucleotide was incorporated and a base was called. In essence, each well works as the world's smallest pH meter. The process is repeated every 15 seconds with a different nucleotide washing over the chip. For example, cytosine. A polymerase incorporates the C nucleotide in the DNA strand if a complementary G molecule is present. If the nucleotide is not complementary to the next base, no ion is released, no voltage change is recorded, and no base is called. If there are 2 identical bases next to each other, 2 nucleotides are incorporated, the voltage doubles, and the chip will record 2 identical bases called. This process happens simultaneously in millions of wells. That's why it's often described as massive parallel sequencing. The ion chips help you scale the workflow to your research needs so that you can run both small and large-scale projects without the need to change platforms. And the semiconductor approach helps you implement a significantly faster NGS workflow. There are many possible research applications with NGS, from whole genome sequencing to identify association of genomic variation with different diseases, to targeted NGS, where the whole power of NGS is used to interrogate a defined number of targets with possible significance in the pathogenesis. Ion Torrent technology from Thermo Fisher Scientific harnesses the power of targeted NGS to make sequencing faster, more scalable, and more accessible than ever before.
If you have any question, you can ask to our team also, or the Ankit also. That we will also use this technology. So, hands up, clear. Okay.